Vanakkam. Welcome to this course on classics in neuroscience. The past few videos we've been looking at the physiology of the action potential or the conduction of nerve impulses through neuronal cell membrane. In this video we will be looking at recording of local circuit activity and graded potentials, crucial seminal contributions of Alan Hodgkin and Andrew Huxley and the historical background to that, and how the discovery of the squid axon essentially constituted a breakthrough for the electrophysiology era that was to come. So, Hodgkin and Huxley, what they did, improving our understanding of the neuronal action potential. Alan Hodgkin and Andrew Huxley were two geniuses who essentially contributed so much. Their contribution to neuroscience can be compared to the contribution of Crick and Watson to biology. So much is their contribution. So, these are two geniuses who collaborated, who combined biology and physics to understand how the neuronal action potential worked. Excellent description using their model. Alan Hodgkin was born in 1914 and he went to Trinity in 1932, where he had the opportunity to study with many brilliant students and also study under the tutelage of many brilliant professors. They were able to meet with brilliant people like J.J. Thomson, who discovered the electron, Ernst Rutherford, who described the atom, Hardy, the mathematician, of course, Edgar Adrian, the physiologist. So, Hodgkin was especially fascinated by the work of Keith Lucas and the lab, Edgar Adrian's lab. That Hodgkin practically read all the papers of Keith Lucas and also read the review by Osterhout, about whom he says the following. I am going to read from the text. I had also read the excellent review by Osterhout 1931 on physiological studies of large plant cells and was impressed with the evidence obtained by Blinks in 1930 for an increase of membrane conductivity during the action potential of Nutella. It seemed to me this crucial piece of evidence was lacking in nerve. So, fundamentally there is a change in membrane conductance. This is happening even in the animal cell, but at that time this was not obvious, but this was first described in plant cells in Nutella by Osterhout and Blinks, two different papers. So, he got interested in exploring this further, and this was his PhD work. This then became his life work. This is not the only example. There are some scientists who start excellent, exciting topics in their PhD and go on to do the same thing for their entire career, because it is a topic that is deep enough and it is a topic in which they have made the seminal contribution and they have a head start over others, and they maybe just love that topic. This is not the first time. There are also other scientists who have done this. So, in the good old days, what was required were chymographs, so equipments that could record information on paper strips, chymographs, some measuring equipment. So, all that mattered to him was that he has access to good quality equipment. So, he describes his lab life in the book. Please do check that. So, Hodgkin's work was inspired by early 20th century scientists such as Bernstein, Hermann, Kramer. Andrew Huxley, meanwhile, was a strong scientist in his own right. He had a strong foundation in physics and mathematics, and then because he was fascinated by physiology of how neurons conduct signals or conduct nerve impulses, he started studying this topic. He is also famously related to the other Huxley, Thomas Huxley, who was his grandfather, who had this opportunity to frequently speak about. My grandfathers used to say this kind of thing. So he was greatly influenced by the fantastic, vibrant scientific community in Cambridge, and he jumped from physics to physiology on mysteries of neuronal function. Obviously, when these two geniuses meet, some wonderful things are supposed to be the outcome. So their early endeavors laid essentially the foundation or the groundwork for creating an entirely new understanding of how neuronal signals are transmitted across cell membranes. So meanwhile, Hodgkin went on a year-long sabbatical or a study tour to the United States, where there was this fancy equipment. There is the CRO, cathode ray oscilloscope, and the people who were developing such excellent instrumentation and electronic amplifiers that were present. And when he described his work and his results, people there, especially Erlanger in St. Louis, expressed total disbelief of subthreshold electrical activity, and also that local circuit current based, that is conduction through local circuit currents. These were, people were highly skeptical about these two concepts. In the meanwhile, in 1936, J.Z. Young described the use of squid axon because its size, the diameter was 1 mm. 1 mm diameter is huge. By neuronal standards, 1 mm diameter neuron is huge. This is a very big neuron. So this made it an ideal candidate, an ideal model system for performing advanced, intricate measurements of action potential. So this discovery, this identification by J.Z. Young proved absolutely crucial, absolutely pivotal moment for the field of electrophysiology or neurophysiology that was to come. Of course, there was advancement in technology that Hodgkin learned about in 1937. I just mentioned these electronic amplifiers, CROs, that is oscilloscopes. So this observation of electrical activity visually in the oscilloscope was possible in the 1930s. So their work with that is Hodgkin and Huxley's work with the squid axon essentially revealed the dynamic changes, very quick changes that happen in membrane potential that essentially constitutes what is now called as the neuronal action potential. So you have this so this is what is shown here is the picture of the neuron. So that is the cell body, and this is the nucleus, and there is this myelin sheath with nodes of Ranvier in between, and you insert a micropipette for measurement and have electrodes outside, and then you make measurements. With this we come to the end of this video. In this video we looked at how local circuit currents and graded potentials were being recorded, early attempts to record this, and the seminal contributions, the historical background of Alan Hodgkin and Andrew Huxley, and how the identification of the squid axon happened to be a crucial or a pivotal moment for the field of electrophysiology or neurophysiology. With this we come to the end of this video. Thank you very much for your attention.